My name is Bobby. I actually have two positions. I'm a neuroscience researcher at Argonne National Labs. This is one of the national labs in the DOE national lab system, like Los Alamos, Oak Ridge, et cetera. And I'm also an adjunct assistant professor in the Department of Neurobiology at the University of Chicago. I think I'm actually literally the first neuroscience person, empirical neuroscientist, to be hired specifically at Argonne, but probably at any of the national labs. And so what I like to say at this moment is that, by definition, makes me the best neuroscience researcher at Argonne. And as my family likes to point out, by definition, that also makes me the worst researcher in neuroscience at Argonne. So I'll let you guys use this talk to figure out which one it could possibly be. One of the things I also learned from my postdoc mentor, I've only been a sort of adjunct professor for about a year now, but one of the things my mentor sort of drilled into me is what's important about being a professor is that you have to profess something. It's not sufficient to just present data and present conclusions. It's important to have worldviews, because that actually helps you figure out what your research should be. So what I'd like to do on the next slide, just very quickly, is tell you what I profess, sort of what I believe, and I suspect this will help sort of guide the rest of this talk. So there are kind of three things that the lab believes. The first, and these are potentially controversial, so push back either now or after the talk. The first is, I'm a big believer that technology is the key to advancing biological insight. By that I mean, lots of people have ideas of the brains. In fact, every time I talk to anybody, it doesn't matter what field they're in, they always have a view about how the brain works. And then they always think their brain is pathological for some particular reason, but it's never the ideas that actually advance us. It's always ways to look at the brain or record from the brain in a way that we've never done before. So that's my first position. The second thing is this talk will only be about controls. There won't be any experiments in this talk, because I actually think that the interesting bit is the control, is to actually figure out how the normal functioning brain works, and that's the best way to do that is by looking at the normal functioning brain, as opposed to always comparing it to some experiment. And the last piece is that this is primarily a talk about anatomy. Here's a quote from a very young neuroscientist, Sigmund Freud, who gave up neuroscience, I learned, to decide to do psychoanalysis instead, maybe because it's easier, but his quote is that anatomy is destiny. And I would argue that there are multiple ways to look at the brain, the functioning brain, the dynamic brain being some of them. But in reality, what we care about is how the brain is structured, because it's that structure that constrains our models for how the brain works and actually reveals possible functions that we would have never guessed simply just by looking at the function of the brain. So just to give you an idea, particularly about that first point, I just added this slide, which is not necessarily the best way to judge great science, but it's one way to judge great science, which is the list of all of the Nobel Prizes in the last, I would say, about 100 years. And the way the list works is you can see the people who won the prize, the year they won the prize, and what's important for me is what comes right after that, which is what they had access to technology that no one else or very few of their colleagues or collaborators had access to. And literally having access to this particular kind of technology gave them the insights for which they won these prizes. And my favorite, although I won't talk more about it, is Erlanger and Gasser, who won the Nobel Prize for recording compound action potentials in the nervous system. And they were able to do this because the lab next door to them was some physicists or electrical engineers who had been working with some new device called an oscilloscope. And they said, geez, that's an interesting thing. Can we use that in the brain to record neural activity? And I think that, along with everything else, is sort of the evidence that I use to think about how technology leads biological insight as opposed to just ideas or hypothesis. Just on the next slide, what I'd like to do is talk specifically about one of these guys, Santiago Ramon y Cajal, and use him just to give you an idea of how I think about brains. And the best way to do that is to sort of see this in a kind of original picture of Cajal that he did in 1903. This is a picture of, I think human cortex, actually, at this point. And Cajal had access to two technologies, which very few of his collaborators did. The first was a stain invented by his frenemy, Camillo Golgi, called the Golgi stain. And what was amazing about the Golgi stain was that it labeled neurons in their entirety, their dendrites, their cell bodies, and their axons, but it labeled only very few of them, maybe less than 1% at some time. And that's the first. The second thing Cajal had was he was one of the first people, I suspect, to use a compound light microscope, a real microscope with objectives and et cetera, to look at brains at super resolution for that day and time. And just doing that, I just want to list the three things that Cajal is famous for, just so we're all on the same page. The first is called the neuronal doctrine, which is kind of amazing to think about now, but at the time of Cajal, people thought the brain was made of just one cell, possibly electrically coupled, but Cajal was able to show with these diagrams that no, the brain is actually made of individual cells, which we call neurons, and that they communicate with each other over empty space, a neuronal connection, which we call a synapse. That's the first thing. The second thing Cajal discovered is called the law of dynamic polarization, which you can also see in this picture, which is that, by the way, here are 1, 2, 3, 4 neurons. Inside of them is the nucleus. Inside of them is the nucleolus. And these little white spots, just if you're curious, are Cajal's whiteouts, as if someone would go back and check this picture 100 years later. So the law of dynamic polarization, you can see, is that these cells neurons aren't symmetrical when you look at them from all different sides. This is different than from lots of other cells that make up organs in our body, liver cells, kidney cells, skin cells, et cetera. They have a certain symmetry to them, but neurons don't. In fact, they're divided into at least 3 compartments, simply, to keep track of for now. One is called the soma, which has the nucleus in it. The thing that Cajal here is labeled brown that goes up of it is called the dendrite, or the dendritic tree, because it looks like a tree. And the thing that Cajal labeled in a different color here in black is called the axon, right? And so I'm enough of a narcissist that when I give this talk, I say, look, if I had access to the Golgi stain, if I had access to the compound microscope, it doesn't seem that hard to look at this stuff and say, yeah, neurons are not symmetrical. I know it's fundamental, but it doesn't seem that hard to do. But there's one thing in this picture that I think is truly genius, not the letters. And if there are any students, I don't know, sorry, or someone else, does anyone want to guess what's amazing about this picture? The arrows, right? Unfortunately for neuroscientists, there are very few arrows in the nervous system, but the fact, and what Cajal means by these arrows, is he's actually tracking information flow in the nervous system. What he's saying is that information flows up an axon of one cell into the dendrite of another, and out the axon of that into the dendrites of the other. We use words now like neural networks in terms of machine learning and algorithms. This is one of the first examples of a neural network. And what's really important for me is that this is dynamic information. He's talking about how information flows in the nervous system before we had any clue that the nervous system did anything like this, that it had electrical signals, that it passed information. And what's even more amazing is he deduced this from purely static pictures. He didn't do time-lapse. There was no capability to do time-lapse at that time. So he took a series of static images and inferred dynamism from it, and inferred this dynamism decades before we were even aware that the brain processed signals like this. And I think this is fundamental, and because this is a talk about anatomy, I use this point to leverage this idea that you can infer dynamism correctly from static images, if you're smart enough, like Cajal. The downside for us as neuroscientists, and here I've taken a title of one of my favorite books from a South American author, is that our worldview has not changed substantially in the 100 years that from Cajal. When we still think about brains, here's a review by Linas and company, where instead of these more accurate neurons, we have circles, et cetera. But I'm going to make the argument that there's no way Cajal is right. There's no way Cajal inferred all of this correctly for this simple reason, that he only labeled 1% of all of these neurons that are present in this picture. One way to look at this picture is to actually think that there's more information missing, probably an order of magnitude, if not more information missing in this picture than present in this picture. And we as neuroscientists have been happy to keep that kind of sparse view of what the brain actually looks like. And the goal of my lab, in some ways, is to fill in these details, is to fill in what it would look like if we actually knew what every neuron and every connection was. And if we could then constrain our models of brains better. Now, this is not a new idea, and in fact, once in the history of neuroscience, this has been done. And it was done for this particular animal, which is called C. elegans. And in case you don't know about it, it's a roundworm. And what I find amazing about C. elegans is that it has exactly 302 neurons. In fact, every C. elegans born, to be born, ever born, has that exact same number of neurons. And starting in the late 70s and moving into the early 80s, a bunch of scientists led by John White et al. Actually mapped all the neurons, all 302 of them, and all the connections within this particular animal. Now, on the next slide, I'm going to show you one representation of what that connectivity map looks like. But I'd like to emphasize one more time that what you've just watched for the last 10 seconds is probably about 99% of all the behavior that C. elegans does. Meaning the vast majority of its time is spent sinusoidal forward, sinusoidal backwards. And I'm glad I timed this perfectly. The little ones do exactly what the big ones do, meaning that there's very little developmental change in the behavioral trajectory of this particular animal. At some level, it's a fairly simple animal. I think there's an argument to be made for a fairly complicated, that's true, right? But when you look at the wiring diagram, which is the connectivity diagram of this particular animal, this is what it looks like. So let me just take 1 minute to explain this. You might see a whole bunch of little dots here, red dots, blue dots, and green dots. If you were a quick counter and counted all of them, there would be 302 of them, because each of them represents one neuron in C. elegans. And these gray lines, which I hope you can see, which emanate from each of these dots and go to the other dots, is the wiring diagram. Is how every one of these neurons connects to every other one of these neurons. And I am interested in doing this for perhaps animals that have more interesting behaviors or different behaviors. And I think it's a fair question to say, okay, these guys did this in 1989, that's almost 20, 30 years ago. Why hasn't it been done for bigger animals, bigger brains, animals with more behavior? And what I'd like to do on this next slide is kind of give you a potential reason why. So on this slide, which is from a review that I wrote with my postdoc mentor a long time ago, I just want to describe the axes. So these are logarithmic axes. On the x-axis, we're going to talk about resolution, right? It's going to go from 1 nanometer per voxel all the way up to 1 millimeter resolution. And on this axis, on the y-axis, which is not logarithmic, I just have data sizes. How big would a data size be if you use this resolution over a particular brain? Now, we're not going to talk about the entire brain. What we're going to do is, just to make the case a little bit easier, I'm going to talk about a mouse brain. And a mouse brain is on order 1 centimeter cubed, right? Centimeter by a centimeter by a centimeter. And I'm going to describe to you how much data it would take to take a small fraction of that mouse brain, 1 millimeter cubed, 1/1000th the volume of the mouse brain. And of course, this is a pretty easy graph to visualize, but let me just show it to you anyway. And so what I've done here is just show you the different imaging techniques and their resolution, and what it would take to, how much data would it take to collect an MRI of a mouse brain is on order kilobytes, depending on the resolution that you use. But we and other people have found that if you want to accurately trace every connection inside a volume of brain, you're going to need something like 10 nanometer resolution. And then, if you just go up the axis, you'll see that a mouse brain could be a millimeter cubed of mouse brain could be a petabyte of data, 1,000 terabytes of data, which isn't that far off, and this is slightly dated, about all the data that lives in all the US research academic libraries in the world. And that's why it hasn't happened yet, because we are waiting for Moore's law and data analysis to catch up with us. And this is true for a mouse brain, but it's 1,000 times more true for a human brain, because that's how much more volume a human brain has than a mouse brain. So I'm interested, of course, in doing the whole mouse brain. But this talk isn't going to be about the whole mouse brain just yet. This talk is going to be about how because we cannot do the entire mouse brain at this resolution, how we're going to be able to move up and down these resolution scales with 2 different imaging techniques to analyze brains at different resolutions. Does that make sense, everybody? I'm going to just take a pause. Okay, good. So the 2 techniques we're going to talk about is X-ray microscopy, specifically synchrotron source X-ray microscopy, and automated serial EM microscopy. And I've just put in there approximate resolutions on this graph. And the goal for me is to map all complete brains at the level of X-rays at micron to submicron resolution, and then take the exact same brain, find the regions we think that are interesting to analyze at connectivity, and analyze those with electron microscopy. Does that make sense to everybody? Cool. And the key, of course, to doing that is to coming up with a way to prepare our brains so that they're amenable to both of these modalities. Great. And how do we do that? And so just let me spend 1 minute so you guys know what the workflow is. So obviously, as this slide demonstrates, we only work on adorable animals. The more adorable, the better. What we do is the animals are fixed with aldehydes, very similar to what dead bodies are fixed in, glutaraldehyde, paraformaldehyde. Brains are extracted from these animals. They're thin sliced. This is actually slightly old. We do the entire brain. And then these brains are stained with heavy metals, osmium, uranium, and lead. And they're stained with these metals because they provide contrast in the X-ray and the EM microscope, the way fluorescent molecules provide contrast in optical light microscopes. Then the last piece we have to do is we actually have to embed these samples in plastic. And the reason we have to embed these samples in plastic is that the trade-off for EM, for electron microscopy, is that although it gives you exquisite resolution, it has terrible penetration. It doesn't penetrate very deep into your samples because electrons scatter. And in fact, even if you had a sample that was 1 micron thick, the electrons that are entering the top of your sample will have scattered away by the time they reach the bottom of your sample. So to really analyze these samples, you have to thin slice them. You have to slice them where each slice of brain off of this plastic cast is on somewhere between 50 to 30 nanometers. And the idea is you collect all of these slices for EM, you take a picture of each of them, and you put the whole thing back together with nanometer resolution. Does that make sense? Awesome. So right now, though, although I've been talking to you about EM, that's the second part of the talk. The part that I'm going to talk about now is X-rays and what we can achieve from X-rays. So what I'm showing you on this slide is this ring that you can see part of. This is the advanced photon source, which is at Argonne National Labs. And basically, it's a synchrotron. Steve, you're going to let me know when I mess up this physics, right? That accelerates electrons around this ring, and as these electrons bend, they give off high-energy X-rays. And these high-energy X-rays have resolutions of 1 micron or less than a micron, but like the X-rays that you have in the medical community, like medical CT, they penetrate deep into your sample. I don't have to thin slice the sample like I do with electrons. And basically, the way it works is I take that same plastic sample that I showed you on a previous slide, I put it on a rotation stage, I shine an X-ray through it, I rotate it a degree or less than a degree, I shine it again, I rotate it again and again, and what you get is a series of what's called sinograms, frequency space of what happens to each of these images, and then you solve the inverse problem by taking these sinograms and turning them into an image stack. Probably there are questions about that, but I'm not going to belabor that point, and I'm happy to talk about it afterwards. So what does this actually look like when we take brains and put them through the X-ray scan? So what I'm showing you as a sort of full 3D volume is a chunk of mouse brain that goes from thalamus all the way to somatosensory cortex, from cortex all the way to thalamus. This is a distance of several millimeters in the mouse, and I'm going to show you what the X-ray data of. So this is a block of tissue that was prepared, like I told you previously, osmium stained, uranium stained, and embedded in plastic. And the dimensions of this particular sample were about 6 millimeters in this dimension, 1 millimeter in this dimension, and only 0.5 millimeter in this dimension. So let me just show you what that data looks like. So now what we're seeing is, of course, not the actual volume, but we're actually slicing through it. And in fact, I have a slightly higher mag view of this. So just to give you guys some kind of, here's the magnified, that red box is the magnified view here. There's a couple of features that I'd like you to see in this data. The first is you'll see little circles appear and disappear. Those are the cells that are found in this part of the brain. So neurons, glia, et cetera. Then you'll see a whole bunch of squiggly lines that traverse this image. Those are that's the vascular system, all the blood vessels that are found. And then what you might see are these huge white sheets that move back and forth. These are the myelinated axons. These are the long distance connections in a brain, and they're called myelinated axons because the brain insulates the electrical signal that's found in them just the way we insulate our wires. So this scan took about 4 minutes to do, and in those 4 minutes we were able to find every cell, every blood vessel, and most of the large myelinated tracts. And what I want to show you And the next slide is sort of our way of automatically extracting, segmenting those features. So this is the work of Eva Dyer, who was a postdoc in Conrad Curting's lab, who now has her own lab in Georgia. But what she did with this data was give a different color to those three different things that I was telling you about. So I believe in this case, the red is the little cells that appear and disappear, the green is the vasculature, and the blue are the large myelinated axons in this particular data set. And very quickly, you can go through from volumes like this and find every neuron, every glia. Here's about 50,000 cells in a particular volume of brain and all the vasculature associated with it that we were able to reconstruct with this particular technique. And I just want to use this moment to kind of show you the equivalent Cajal region. So here's what Cajal labeled in his particular data set. And here's some of what he's missing, right? And my view is that knowing this information constrains this data in ways that Cajal couldn't give us. But we don't really want to do this for small volumes. We want to do it for the entire mouse brain. So what I'm showing you now is that an entire mouse brain, 1 centimeter cubed, imaged at something less than 1 micron resolution. The image itself is downsampled, so I can't show you all the little cells and the little myelinated axons and et cetera, but they're there. And the reason I'm interested in this is I want to be able to count every cell in this brain. Because when I talk to people and they say, well, how many cells are there in the human brain? Well, then we have to say something like, well, there's 100 billion cells. Plus or minus, I don't know, 50 billion, plus or minus 60 billion. That's embarrassing, right? If we're going to treat this as a serious systematic study, we should know answers like, how many cells are there? How do they vary? How do they vary in young animals, old animals, diseased animals, et cetera? By the way, how does all the vasculature and all the myelinated axons vary in these particular data sets? And although this is still a mouse, here, I love this. This is where all the streaks of myelinated axons in the interior of the brain. Because ultimately, with X-rays, we won't be able to stop. We don't have to stop with a mouse brain. We're going to try to do our first macaque primate brain exactly like this. So we could compare all the cells and the myelinated axons and the tracts and the vasculature between species to see how many different ways evolution has come up with answers to these questions. So here again is work by my graduate student Han-Yu Lee, where he's taken that grayscale image data set and now tried to extract all the blood vessels and all the myelinated axons from this particular data. One of the reasons we're really interested in this is this particular brain, in fact, this exact brain, before we did the X-ray analysis, we did MRI analysis, meaning we ran it through an MRI scanner. We let them do their T1, T2, T-star, et cetera, et cetera, et cetera, and let them do their version of myelinated axons. Let them find the myelinated tracts, the DTI, DSI. No one's ever ground truth these guys. No one actually knows whether when they say these axons go left and right and then make a U-turn, what does that actually look like? So by comparing the same brain with the MRI to the X-ray with an increase in resolution of about 10,000-fold, we'll be able to ground truth and vet these MRI algorithms. That's one version of how we see this data going forward. In the next slide, another thing I'm extremely interested in with Greg, who's a postdoc in Marcus Kornforst's lab, is this is an entire butterfly brain. It's about 3 millimeters cubed. And one of the things I've learned about butterflies is that they're actually a model system for studying genotype to phenotype expression, specifically at the level of what their wings look like, right? The colors of their wings, the shape of their wings. People know tons about this. What people don't have no idea about is the genotype to phenotype expression at the level of brains. What do different species of butterflies look like when they have different functions, when they have different evolutionary trajectories? This data takes us about 3 minutes per butterfly brain to collect. And so we've now collected, I think, 20 brains from 2 different species that have been extremely well genetically characterized. And I'm interested in an unbiased sort of QTL screen of brains relative to the genetics. So anyway, these are just some ideas of the kinds of X-ray work that we're doing. I'll point out everybody who did the work and later on in the talk, like a good PI. I'm going to end this X-ray part of the talk right now. I'm happy to go back to it later, but I just wanted to see if anyone had questions about the X-ray part. Please.
So do you expect in your 2 butterfly model, I mean, as you're thinking, are you going to see different numbers of cells, different shapes? I mean, what?
Yeah, so we are open to this. So ultimately our goal is to take this raw data, segment everything, and then essentially use algorithms, machine learning algorithms, to tell us what the difference is, as opposed to coming in and saying, we think it's shape size or clustering or et cetera.
Could it be the same?
Yeah, it'd be great. I mean, at the level of our analysis, it could be the same. And here's one thing about butterflies that I find kind of interesting. They don't respect species definitions the way other biologists do, butterfly biologists, in the sense that you can have two different species that are called two different species, and they could still mate and produce viable offspring. Why that is something left between you guys and butterfly biologists. So of the 20 brains that we have, I think like 7 are from each parents, and then we have 7 of the children as well, and the genetics for all 3 of them to try to ultimately map this out. Please.
So similar question for the mice. Have you done several mice, perhaps of different strains?
Yes, so we have done 3 mice of the same strain. Sorry, that's a no. No, we've only done 3 mice of the same strain because we want to get some N numbers going before we start comparing.
Sure, but assuming the same strain, what kinds of variability do you see in individual brains of the same strain?
Yes, so what is stopping us is really, and I'll talk about this, is there's on order 100 million cells in the mouse brain. We're trying to solve the problem of how to segment those with the highest specificity and sensitivity that we have. Once we do it, they're going to start applying it to the data set. So we will have an answer. My suspicion is it will be at the level of numbers of cells and clustering of cells. So for the rest of this talk, I'm going to subtly switch over to our approach to automated serial electron microscopy. So just to give you this idea, here is that same plastic block that we theoretically did X-ray on. Because the X-ray is non-destructive, we can take that same block and put it through our pipeline for electron microscopy. The first is we do a whole bunch of ultra-thin slices. I told you why, because of the scattering nature of electrons. Then we take an EM image of each of those slices. We align them post hoc, the images from one to the other. Then we look for signatures of neurons in each individual slice, the borders of them, and then we connect that all together to make a 3D object and track them. Does that make sense to everybody? So as soon as we started thinking about this, the first thing we realized is that this part, this serial ultra-thin slicing, was currently done by hand. Meaning 9 years ago, when you wanted to collect a bunch of EM sections, you had a human being who would collect each or 3 sections at a time, right? And a millimeter cubed of mouse brain at the thinness that I would like is 20 to 30,000 slices. An entire mouse brain is 300,000 slices. And of course, we don't want to do this once. We want to do it multiple times. So we realized that the first thing that we had to do was automate this process. And if you didn't automate this serial ultra-thin slicing process, then everything else downstream would have been useless. So what I'm going to describe to you in the next slide is our approach to how we automated the serial ultra-thin slicing process. So in this CAD diagram, I'm just going to spend 2 minutes describing it. Here again is that same plastic block. I forgot to mention this. The brains are turned black because the osmium stains them black. So that's supposed to be the brain. That brain is attached to a chuck, and that chuck is attached to a cantilever that moves up and down like this. And it also advances in X nanometer steps, 30 to 50 nanometer steps. Just sort of opposed to that block is a blue rectangle. This is called the diamond knife, and the reason it's called the diamond knife boat, is that the tip just next to the sample is actually a piece of diamond. A piece of diamond that has a radius of curvature of like a nanometer, which is another way of saying it's like super sharp. And what happens is as the block advances, slices get cut by this diamond knife and they float on the surface of this water. Up till now, everything that I've been describing to you is standard serial electron microscopy that's been done for decades. Our addition to it is this Rube Goldberg-esque taping mechanism, which actually sits in our knife boat and automatically collects section after section after section after section. It's very important for me to tell you that this has been patented and licensed. So if any of you decide to buy this instrument because of this talk, the way Harvard runs patents, I stand to make dozens and dozens of dollars from this. So what I have on this slide now is actually the machine working in real time. So there is that block that's moving up and down. Here is my diamond knife boat. Here's the diamond knife. As the block hits, a section is formed, which is in shade, so it's hard to see. But you can see that the previous section gets pushed onto our conveyor belt. And the message I'd like to leave you with this is a kind of a message of boredom, right? Meaning the way this works is that you set up your sample, you go away, it just does this again and again and again reliably, and you come back and you have 10,000 EM sections or 5,000 EM sections. The record, not in my lab, but in my postdoc mentor's lab, is I think now 32,000 sections using this approach. So we're at the level of millimeter volumes, and we hope to expand this even further. So just to continue with how this approach works, you take that, so now you have a tape with a whole bunch of sections on it. We actually cut them and adhere them to the surface of a silicon wafer, and this whole thing is going to go inside a scanning electron microscope. And we call the collection of wafers that contains a brain, let's say, an ultra-thin slice library. So what I like about this wafer method is I think this is why CDs beat tape, tape for audio. This allows you to have random access to any of the particular samples. So if we kept it on the tape, if we were on slice 1 and we wanted to get to slice 10,000, we'd have to zip the tape all the way in this direction, zip the tape all the way in the back direction. Whereas if we have it like this, we have random access to this entire data. And I'll tell you why that could be useful later on. Now, because we put these on tape, and that tape is way thicker than a micron, if you guys have been paying attention, there's no way that we can do transmission electron microscopy on this data set, because the electrons are going to scatter through the tape, and if they don't scatter through the tape, they're going to scatter through the silicon wafer that we attach to. So the data I'm going to show you doesn't come from transmission electron microscopy, which is the kind of traditional version of electron microscopy. It comes from something called reflected or scanning electron microscopy. What we found, and many other people found, is that the data from scanning electron microscopy looks identical to the data from transmission electron microscopy. And I'm going to prove that to you in the next slide, the next 2 slides. So here's what that looks like, scanning electron microscope. So let's actually look at some of the data. So here is a section of a mouse brain that goes from the very top of the brain right here, the top of the cortex, all the way down to the bottom of the cortex right here, right where you could see myelinated axons entering. This is a very low res image, but even at this low res image, you should be able to make out the little cell bodies that we saw both in the Cajal diagrams and in the X-ray data sets. But what I'm going to do is just kind of zoom up. I'm going to zoom up on that region right there. So if you can keep those cluster of cells in your mind for a second, you'll see here they are. So once again, I'm just pointing out 1, 2, 3, 4, 5, 6, 7 cells. You might be able to see the nucleus inside of this cell, and all of this gray mass, all of this stuff that surrounds these cells is profiles of axons. Like, this is how complicated the brain is, and it looks nothing like Cajal or, frankly, fluorescence images of how we see the brain. So here's what I'm going to do. I'm going to zoom up on this region because we're nowhere near our full resolution, so I'm going to zoom up there. And now, actually, you should be able to make out the border, the plasma membrane border of this particular cell. Inside of that is the nucleus. And you might even start to see intracellular organelles, like here some Golgi. These things that are dark are the mitochondria. And here is actually a myelinated axon with a black ring around it, which is the insulation. Excuse me. So I'm going to zoom up again. In fact, I'm just going to zoom up to our full resolution. So this is actually what most of the brain looks like, this amount of material. So let me just spend, again, 1 second describing this image to you. So here, what I'm tracing with my laser pointer, I hope you guys over there can see it, is the outer membrane of a cell. Now remember, these are 2D linearizations of 3D objects. It's a single slice, right? So you're only seeing what is essentially a contour, a profile of a cell. Now, I happen to know that this is an axon as opposed to a dendrite. The reason I know that is because this is filled with all of these little circles. They're not circles, they're spheres in real life in 3D. These are synaptic vesicles, right? And if you look, this profile is cut off, but there's a profile of a cell right next to it that abuts it. There's a dark density between these. That's actually called the postsynaptic density, and this is a synaptic connection, a synapse between this axon. Remember, axons send information, and this dendrite, which receives information at this position in space. And if you're keeping track, here's another synapse. Here's the profile of a cell. It's an axon. It's filled with all of these little vesicles. And here's the profile of the cell right next to it. There's a dark density between it, and that's the synapse. One thing that's useful to say is that we actually have enough resolution to kind of to sort of characterize the pleomorphy of these vesicles. What do they look like? What size are they? Are they pointy? Are they not pointy? And that's important because not us, but people who do EM for a long time have used this stuff to be able to distinguish different kinds of neurons. So you could tell the difference between an inhibitory cell and an excitatory cell based on the size and the pleomorphy of these synaptic vesicles. Please.
How many microns across?
Great question. I apologize. These circles are on order 30 to 40 nanometers in diameter. So apologies, I should have said that. So now what I'm going to show you in this next slide is not a single image, but a stack of images that has been aligned together. So again, so that's what you're seeing now. In this case, you're seeing little cells appear and disappear. You're seeing the blood vessel, here are blood vessels just like we saw in the X-ray and the Kahala diagram, but you might also notice there's a lot more squiggles, right? These little white squiggles are the dendrites, apical, basal of all of these cells. You could see little black myelinated axons squirting through. Remember, there is no motion or movement. Nothing is moving in this sample. I wish that would have been a much better result than we have. The reason you get this impression of movement is that you're just moving it through space, and what you're seeing is movement through space as opposed to real movement. I've gotten that question a few times. Now what I want to, this is of course a downsample of the raw data. So let me just show you an even smaller version. So here I'm going to describe the process by which we go from raw grayscale images to 3D segmented objects. And just before I start, just so we're all on the same page, here again is the profile of an axon filled with synaptic vesicles. Just next to it is a dendrite, specifically something called a dendritic spine. And here's the PSD that suggests that there's a synapse between these two. But as I was saying, we don't actually have a kind of a single frame. We actually have a volume of EM, right? And the goal is to find one cell and find it again and again from section to section to section. It's pretty easy because it doesn't change very much, because it's only 30 nanometer thickness, and then once you've done that for every slice, you can produce a three-dimensional reconstruction. So here's an orange dendrite with a blue axon. Is it making a synapse? Yeah, we can go back to the raw data, and again, here is our signature of a synapse. The profile of an axon filled with synaptic vesicles and a little dendritic spine, the postsynaptic density, and there's the connection, of which there's probably 99,999,000, la, 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 et cetera. And the goal is to get those, is to get the ones that we're missing, right? And as an exercise in two things, first in figuring out how complicated the brain could be, and second in figuring out what was my capacity for suffering, we decided to take a really small volume of brain and trace everything inside it, just to get an idea of how complicated the brain could be, right? And what is it that we're missing? And so the volume that I'm about to show you is 1/1000th the volume of a mouse brain, which is 1/1,000,000th of a mouse brain, excuse me, which is 1/1000th the volume of a human brain, and took me about 6 months to trace. And there's a reason I'm putting that 6 months in. It's literally smaller than a grain of sand. So here again is that same EM data set. This time it's now multicolored because a human being, some crazy human being, me, went through each, and we just circumscribed the volume. You see these weird little cylinders that appear. That's just to give me a moment to stop. But basically what I did was just trace the same neuron, the same color, from section to section to section to section. Okay, so this is smaller than a grain of sand. How complicated is it? I'll show you the three-dimensional reconstruction. Here again are those cylinders, which is literally smaller than a grain of sand. And because it's so packed with stuff, I'm going to explode it for you so you could see how complicated it is. So in this particularly small volume, which represents nothing in the brain, there are thousands of different axons, hundreds of different dendrites that somehow all come together by some rule and make the accurate wiring diagram again and again and again and again. And I'll just say this, maybe people in the audience know, we have no idea what those rules are. One other thing to say is because of work done by lots of people that's not us, it's easy for us to sort of characterize the different components of this data, right? So it's easy for us to see things that are axons and dendrites and things that are neither, these support cells, oligodendrocytes, astrocytes. I'll get back to this category in a second. And then within axons, we can characterize them as excitatory versus inhibitory based on the ultrastructural characteristics I was telling you. And we can also know dendrites that have lots of spines on them are from excitatory cells, and dendrites that don't have a lot of spines on them are from inhibitory cells. So we're not only able to find the trajectory of these things, we're able to classify them into different types. And so one other thing we actually discovered, which I think is actually the result, is that there might actually be rules for how axons and dendrites connect with each other. So let me just tell you what I mean by that. Here is a single dendrite labeled in green, and all of the spines that come off of it. And this is the kind of spine density people should be getting if they were doing spine density. And you see a single blue axon that comes through, and it connects the same dendrite 5 different times, right? Which is surprising to us because given how many axons and dendrite combinations there are, just by chance, you wouldn't get 5 times, you wouldn't put your 5 eggs in the same basket if you had all of these other baskets to choose from. And I'm not going to go through the statistics of this, but it turns out that finding axons that connect to the same dendrite should be a rare event if the connectivity was random. And I know that randomness is the easiest thing for people to exclude, but that's at the level we're at in terms of this kinds of connectivity, and we find this happening again and again and again. So there's something linking this axon to this dendrite, having this axon prefer this dendrite again and again and again. It implies a kind of rule. Now what that rule is, we don't know. Please.
Is there a difference between a sensory and a motor dendrite?
Is there a difference between a sensory and a motor dendrite? I'm not sure exactly how to answer that question, in the sense that there is, if both a sensory dendrite and a motor dendrite were excitatory, then at some level they should look the same. And I would argue in my position, the difference, what makes one sensory and the other motor, is who connects with it and who it connects with.
So it's just the action of the axon, not the dendrite?
That's my particular view. People might say, sorry, is it just the axon? It's the action of both. I'm not claiming that it's the axon, because the axon transmits the information, the dendrite receives the information. You couldn't make it happen without either.
And there's just one package of information that we don't need 5 connections for?
So that's the question. If there was one package of information, why do you need 5 connections for it? I don't know the answer. I don't think I did a good job answering, but I'll try again later. Please.
So that blue axon, does it connect to a different cell type?
Yes,
And
it does.
and my second question is, if you have 5 connections to the green dendrite,
Yes.
yeah, and you have a, I don't know, maybe the blue stuff connects to a red cell, yeah?
Yeah.
If you would have only like the blue axon connecting to 3 has 3 connections to the dendrite, would that then connect to maybe a yellow cell? Could the amount of like connections also define how to which other cell it connects? Does that make sense?
Let me try the first one. The second one I'm not sure of. But let me try the first one. The first one is that in this field of view, although not seen, this axon is connecting other dendrites of other cells. It does not exclusively just connect this dendrite. It connects other dendrites in the field of view. But it's just unlikely for it to connect this dendrite 5 times, when, as I showed you, there are hundreds of dendrites crossing this path that are not visualized.
But my question was, can you see the same pattern, like that when a blue axon connects 5 times to the green dendrite,
Yes.
that you would always get then a connection to maybe a specific cell type, like the blue axon connects then to a specific other cell type?
Meaning, okay, let me just put some numbers in. Let's say there's 7 connections here,
Okay.
right? I think you're saying every time I connect 5 to this guy,
Yeah.
do I always connect 2 to another specific guy?
Something like that.
Yes,
So that
we
you
saw
have
some
something like
yes.
A spatial, so that you can say, I do 5 times, I connect to this cell, the
Yes.
signal goes this way. If I only connect 3 times, I go to that cell and you can.
I think this is true, but our volume was so small that we really can't say. But there's good news coming. Oh, you have one. Please.
What is the white line?
This line, this arrow.
No, the one down there.
Oh, you know what? That's a great question. That's called a scale bar. And I should be telling people what that scale bar is. It's about 1 micron. It just tells you how big something is in the image. That's a great question. So one last piece before I move on to sort of the end of this talk. The other thing I want to tell you is that when you see these Cajal diagrams, the other thing that's missing is what happens in the interior of the cell. So Cajal couldn't actually visualize all the stuff that was inside a cell, right? He could only see the nucleus, maybe, and a little bit of the nucleolus. But because we have all of that information, we can actually produce three-dimensional renderings of cells that are accurate down to every organelle. So what you see here is a neuron that has a glass skin, right? That's the plasma membrane of that neuron. Inside that neuron is a gray sphere. I hope you guys can see that gray sphere inside that neuron. That gray sphere is littered by little black dots. Can you guys see those little black dots? That gray sphere is the nucleus, and that is every nuclear pore that is found inside that nucleus. Not an estimate of the number of nuclear pores, but the actual location and number of every nuclear pore. These things that are orange, these little squiggly orange lines that are found here, is every mitochondria that's found inside this particular cell. The thing that's here that's in black is every lysosome that's found inside this cell. The thing that's here in purple is all the smooth endoplasmic reticulum that's found. And what we are seeing, which I guess is sort of obvious, is that cell types in the brain differ by their composition of organelles. So for example, we find that inhibitory neurons are packed with way more mitochondria than excitatory neurons. Now, to me, there's a potentially easily explanation. It's that inhibitory neurons fire 10 times more often, let's say, than excitatory neurons. So it's not surprising that therefore they should have more mitochondria to support the larger energy demand. But what I'm interested in is, can we continue to classify cell types based on what their internal and their external structures are? I mean, that I think is a new way of thinking about this potential brains, where we don't have to use genes. We can actually use the things that are doing the work to classify the system. In the last couple of slides, I just want to push one more point. If we wanted to do an entire mouse brain at this resolution, it's 450,000 terabytes of data. Now that seems like a lot. Actually, I think that is a lot of data, but at a place like Argonne, it's not a big deal. They're used to these kinds of data sizes, which is why a lot of this work is being done there. Now, just to give you a scale, to do a human connectome, somebody was asking me this at lunch, is about 1.3 billion terabytes. That's a little bit more data, but it's actually not an inconceivable amount of data, because if you calculated how many hard drives existed in 2014, turns out there's 2.6 billion terabytes worth of hard drives that existed. And so I wouldn't even need all of them just to store a human brain. I would only need half of all the hard drives that existed. And that number is going to get better and better with time because of something called Moore's Law. And on the right-hand side, what I wanted to show you is that it's not going to be our inability to acquire the data. Right now, companies like Zeiss are producing electron microscopes that can literally scan gigapixels per second, right? I mean, and the way they do it is right now, it's an amazing physics engineering thing that I'm still trying to figure out. Right now, what most scanning electron microscopes do is they have one electron beam that scans the surface of your sample. What Zeiss figured out how to do was to scan 91 beams at the same time with 91 independent detectors so that you get 91 times the throughput. And by the time I'd made this slide, it was 61 beams, and then it turned out to be 91 beams, and now it's 121 beams. And so this ability to collect the data sizes that we need is not going to be the showstopper. What's going to be the showstopper is who's going to analyze all of that data, who's going to read all of that data. And in fact, to kind of push that point home, I want to show you how pathetic my postdoctoral result was. So,
Bobby, can I interrupt you for 1 second?
Please.
So the numbers you just gave us about the amount of data it takes to store a mouse brain or a human brain, can it be compressed? Can we treat it like a JPEG? So there have to be a lot of things in there that get repeated, and we compress it in some way.
Yeah, I mean, yes. So the answer is yes, but it scares me, man. Like the idea that we're going to go to all this effort. And by the way, this is something physicists are totally comfortable with. Like the people at CERN throw away 99% of the data that they collect, right? But they have a much better idea of what they're looking for versus what we're looking for. So of course there are ways to scale and compress the data. I've just been assuming that we're not going to want to do that because we're too dumb to know what is the actual, not you guys, we're too dumb to know what's the actual thing we're looking for. So that's why I hesitate for thinking about that stuff. But I'm sure there are computer science people who will say, of course you can compress it without information loss, et cetera, et cetera, et cetera. I wouldn't argue.
I say that especially because your images are serial, right?
Yes.
So a lot of the
Yes.
It's in this image too.
Yes, but it turns out that if you skip an image, right, the algorithms have a pain in the ass trying to figure out which were the two sections there. In fact, what the algorithm people want us to do is to go to even thinner sections, to add more Z slices. Not 30 nanometers Z, but 10 nanometers Z, so that the problem becomes trivial for the algorithm. The whole field, as you'll find out, is to appease algorithms. And the reason we have to appease algorithms is here. I told you it took me about 6 months of effort to do that cylinder. Here's a bunch of cells to tell you, and I want to show you how pathetic my effort was. There's that cylinder, man. It doesn't even cover a small fraction of the dendrite of a neuron. And it took me 6 months of tracing, plus some cigarette breaks, but mostly 6 months of tracing to do that. So this is a field that is going to live or die when algorithms can trace neurons, if algorithms can trace neurons as well as humans can do them. Because we did the calculation, it's easy. There are not enough people alive on the planet, I think, ever been alive, working a 40-hour week to trace all the neurons and connections in a mouse brain. It's just never going to happen, right? So the only way it's going to happen is if algorithms do it. And luckily for us, a whole new sort of realm of algorithms called machine learning or neural net algorithms have emerged that try to replicate human behavior, but with the speed of algorithms. And what I'm going to show you in the next slide is some very preliminary results from these algorithms on tracing EM data sets. So once again, here's our whole EM stack. I was showing you a volume like this, right? Which took me about 6 months to do. This was the Harvard supercomputer. This took the Harvard supercomputer about 6 hours, maybe 8 hours to compute. And it doesn't do a terrible job. In fact, it still makes mistakes. Look at this cell that's coming into the field of view. You'll see these weird block segmentations appearing. That's a mistake. And in fact, anytime you see a quick color flash in this movie, it's a mistake, right? And the thing that bothers me about these algorithms is that they're just unbelievably arrogant. But by that I mean, when I teach a human being how to trace, a human being usually gets back to me and says, you know what, this cell, that was pretty easy to trace, Bobby. You don't need to double check me here. But here, when it got really tight, I don't know if I did a good job. Can you please go back and double check me here? These algorithms produce output without any confidence intervals currently, right? So the algorithm is equally confident in every pixel that it has laid down on this particular data set. That's nuts, right? Because for me to know whether there are errors there, I have to then scan through this data set all over again. And that turns out to be as long as actually painting the data set itself. And that was the state of the art about 2 years ago. But what has happened, which is kind of amazing, is that a bunch of commercial companies, private companies, have taken up this challenge. So the best algorithm for tracing EM data sets of brains comes from Google. And why Google is investing $8 million or $10 million a year on doing this is, I have a hypothesis, but I'm probably not going to say it out loud. But the advantage of it is that before the Google result, which happened about 4 months ago, before the Google result, you could trace an axon about 5 to 10 microns before the algorithm made an error, right? Axons travel much longer distances than that. So it was really a non-starter, right? But for that, and then Google published their result. Viren Jain, who used to be a graduate student at Sebastian Sung and worked at Genelia Farms, they can now trace an axon 1.4 millimeters without an error. And that means we're close to the level where we can actually trace out circuits. A millimeter is actually a reasonable distance where you might be able to actually trace out circuitry. Now the downside is I think it requires Google computing to actually make it happen, but they're in the process of making their algorithm less and less computationally intensive. So this is where I think the hope is, at least for this kind of data. And really, I'm just going to summarize at the last couple of slides. What I'm interested in is this idea of some, that we creatively call XM microscopy, where you could take brains and with X-rays do coarse resolution mapping of that entire brain. What do cells look like? Where are they distributed? Maybe what are their dendrites? What are the major pathways between them? And then take that same sample, subdivide it for electron microscopy, and redo the circuitry in the regions that you care about. And we're going to push this until the whole 1 centimeter cube volume is solved. And essentially this is the idea. Take a whole bunch of cells that you got from the X-ray, find the interesting region, and do electron microscopy on it. I'm almost, oh, not bad, I said early. I said late. Yeah, good. I must have talked faster than I thought. I'm essentially, one more question.
How did you choose how to color code each different cell?
It's a great question. I didn't choose. We have this drawing program that I say next object, and it just assigns a random color to it. But it is a fair point that once we know what these cells are, like what they do, we can make the color correspond to what they actually do. That is a really good question. Please.
Correlation between murine and mammal?
What, meaning, what's the differences?
If you do a murine, does it apply to the mammal?
This is a great question for the field, not just for what we do, but one of the things we're absolutely interested in is comparing the differences at the level of connectivity between mouse, primate, and human. The problem with human is it's hard when we get their autopsy, but there's some X amount of time between when that person is declared dead and when that brain is put into fixative. Sometimes, if we're lucky, that's 12 hours. If we're not lucky, that's 24, 36 hours. And by that time, the brain has degenerated to the point where we can't do these reconstructions. But the mouse to primate, we can do, non-human primate. Please.
So are you also concerned about postmortem time? By the time you isolate your brain, that your mapping has disappeared or changed?
Maybe we should be, but I just want to say we don't do anything. It's called perfusion fixation, where we essentially open up the heart while it's beating and directly inject fixatives into the animal. That's not something we invented. That is sort of standard procedure for electron microscopy. Now, it's possible that that has artifacts, but I don't know how else to verify it because I have to do that to do the high resolution EM. So it's a fair point. We know, I think it's also fair to say, we know that aldehyde fixation changes the three-dimensional structure of the brain. We're dehydrating the sample. So when we look at the morphologies of cells, they're probably not the exact morphology that you see in the living brain, but what we think doesn't change is the connectivity, who connects to who. And of course, that's not work that we've done, that's work that other people have done for a long period of time. Please.
So, for animals that learn through their life,
Yes.
I suppose the neural connections would be different between...
Could you speak up?
Well, for animals that learn through the life,
Yes.
the neural connections would be different.
Yes.
And each individual would have a different neural connection.
Correct.
So, I guess what is your prediction in terms of where this is all going to go? Because from taking from one minuscule amount of tissue in
Yes.
the brain
Yes.
gives you this much data.
Yes.
And that's going to be different from a different brain.
Correct. So I think there's not much utility in doing minuscule volumes of brain like this. I think the volumes we want to do are things like on-range millimeter cubes. The reason I keep bringing up millimeter cubes is that there's a fundamental hypothesis that there's something called a cortical column, a column of cells that's a repeating unit in the brain, and that the margins, the size of that is on order millimeter cubed, right? And so the goal is not to do 1 millimeter cube, but many millimeters cubed. But even when we do that, there is a chance that every brain is a snowflake, a neurological snowflake, right? That each of our brains is unique because of the experiences that we have learned, right? I'd be happy to know that. If that was the result at the level of connectivity, done. But I would argue, even with my analogy for snowflakes, there are fundamental things that all snowflakes have in common, right? They have 7 arms, 6 arms, I can't remember, something like that. And there is information on top of that that is unique to each particular snowflake. And I have a simple question: which parts of brains are the same and which parts of the brains change? And I suspect we will get some of those answers by this approach. It could be that we're screwed, right? But if we're screwed, lots of other people are screwed too, because there are many people that work on mammalian brains, and their results, no one says, are unique to that particular mammalian brain, even though the connectivity map is different. Oh, another one, please.
Why is some color and some parts are just gray?
Yeah, because I was lazy. I would have loved to have made this whole thing colored, but then I would have never got this job.
You alluded to the controversial cortical column hypothesis.
Yes.
In the barrel cortex, you can certainly see those
Yes.
very clearly, but I don't see in any of your pictures anything that looks like those kind of cortical columns. Can you answer that question already negatively?
So I should push back a little bit on this. The idea of cortical columns is sometimes different in primates versus mice, There are lots of things that happen even in rats that don't happen in mice. So when you do functional imaging in these animals, rats, primates, cats, you see functional cortical columns where the orientation is in one particular column and in a different orientation. In mice, it's way more salt and pepper. The functional analysis is way more salt and pepper. So since most of this work is being done in mice, it might be that this idea of cortical columns doesn't live in mice. I'd love to say this as an answer, but I suspect we're going to need more data before we can confirm it. Please.
One other question. 2 years ago, Yuri Kuzaki published a paper arguing that everything in the brain was scaled according to a log normal distribution, so there should be, a lot of small, in fact, he said that there were lots of small pyramidal cells and then a long tail of very big ones. I don't see that in your data. Do you say that that's just not the case?
Man, you really put me. Anybody here know Yuri Buzaki before I say? I would say yes. We don't see a long-tail distribution of large cells in our data sets. Thanks. So I'm essentially done. I just wanted to leave you with one more slide and then talk about how I get funded. I like this slide because I always like to end this talk, and I know that there's a certain fraction of people in the audience who think we're nuts, or I'm nuts, and that we're crazy, right? And that we'll never get it to work. All of those things are true. The reason I like this, and this is the quote that John F. Kennedy gave at Rice University around 1961, and he says, "We choose to go to the moon in this decade and do the other things, not because they're easy, but because they're hard, because that goal organizes and measures us, and because that challenge is one that we're willing to accept, unwilling to postpone, and one which we intend to win." I've seen this part of the quote many times, but I recently found this part of the quote. But why, some say, the moon? Why choose this as our goal? They may as well ask, why climb the highest mountain? Why 35 years ago fly the Atlantic? Why does Rice play Texas, I assume, in football? And the part that I not mentioned in this quote, as I read this recently, is that in 1969, when we landed on the moon, the average age of a NASA scientist, a NASA engineer, was 28, 29 years old. That means when JFK announced this, those guys, and mostly guys, unfortunately, and women, were in college, right? When they heard about this particular idea. And what I think is the real benefit of big projects like this are the incidental discoveries. It's possible that we're never going to map an entire mouse brain. In fact, likely, unless that we don't map an entire mouse brain. But I guarantee we're going to find a bunch of interesting things moving along when we think about connectivity. And the thing that I find fascinating is that this is going to change the field, I suspect. Not what we do, but this general idea of collecting brains and mapping them at high level, because it's going to be the final unification between computer science and neuroscience. It's already happening every day. It seems like that these two fields are merging closer and closer together. Here's a huge field that requires massive computer science efforts to actually make it work. And then the reason the computer science want to do it is they hope that they can reverse engineer the algorithms that that biological brains do to actually make our current algorithms and our computer architecture better. Whether that works or not, that's what I'm actually quite fascinated with. Okay, this is the last piece, which is to tell you who pays for all of this stuff. We're funded by IARPA, NIH, NSF, the McKnight Foundation, which is a private foundation, and DOE money through the Argonne Lab-Directed Research Directive. Thank you very much, and I'm happy to take even more questions.
I know some people might have to go, but we can field a couple questions as people want to find out. Any other questions?
Please, I'll throw another.
When we talked earlier today, you mentioned this idea of labeling individual
Yes.
neurons with something that would be electron dense.
Yes.
Are people doing that with cell type specific?
Yes.
So this is exactly what's happening right now.
Yes. So
Go ahead.
I'll just give the credit. Sorry. The molecule is called Apex. It's a P peroxidase. It was discovered by a woman named Alice Ting, who I believe is at Stanford now, and it's essentially like horseradish peroxide. It's essentially like GFP. When you express it in a, you can genetically express it in a particular cell, and then it turns that one cell type completely dark in the EM data sets. And Josh Shanes at Harvard has expressed them in specific cell types in the retina, specific amacrine cell types, I think, in the retina, and then demonstrated that they only express, et cetera, et cetera. Sorry.
So my question is it dense in the X-ray?
Yes.
So you'll be able to find it
Yes.
with the X-ray?
Yes.
And then do your EM just on an area that has a couple of these cells?
Correct. So very recently from the Allen Institute, And I can just show you afterwards. From the Allen Institute, they express this molecule in layer 4 cells that they care about, and we did the X-ray scan of it, and you just see it popping out.
And it's Cree-dependent.
It can be. I mean, it can be whatever GFP can be, I guess, at some level.
So I guess then the idea is, can you manipulate other genes in a Cree-dependent manner at the same time, and then see how that changes the
Sure.
shape of those cells?
Sure. I don't know how to do that, but I'm sure somebody knows how to do that. And then we would help analyze it, but we wouldn't know how to make it. Okay, great.
That would be
Awesome. Please.
So when will some of these data start appearing in publicly accessible databases?
Oh, so all of the data that I showed is freely available, including the segmentation, et cetera, at a place called openconnectome.org. They might have rebranded it neurodata.io. I don't know why people do that, but almost all of the big EM data sets that people have collected are now online. Is that what you meant? Or?
Well, I actually meant it in two places. So at least the ones that I saw there were being mostly graphics files, but maybe I was at the wrong place, but I can talk to you later.
Yes.
But how much of it's going to be available to your average non-informatics person? So yeah, I'm an informatics person. Yeah, I can read some of these big files.
Yes.
Most people here are not going to want to do. They're going to want to see in more detail the kind of pictures that you and the Allen Institute folks show in the public talks.
Yes.
They want to say, okay, can I look at how long these distances are? How far
Yes.
is between the branches and the dendrites?
Yes.
Things like that.
Yes.
When is that going to be available?
So if I'm in charge of it, it's never going to be available like that. No. I just mean I'm not an informatics person, so I don't know the right image formats and the curations and et cetera that are appropriate. Instead, what we would love to do is you give the data to people who are informatics people, and you tell us what's the right way to format and analyze and et cetera, and we'll just do it. But because that's just not my expertise, and I would muck it up pretty bad, I think, if I tried to do it myself. But it's ready. If you're ready, I could give you the data later today, and you let us know when it'll be up.
Any more questions?
Oh, yes, please.
So how are you able to design the types of, because you're sitting on huge mountains of data sets, how are you able to design the types of questions that you would ask that would be able to really mine that data effectively? For example, seeing the 5 dendrites there, you might ask questions on
I see.
on how there's, is there synaptic competition or something like that going on? How are you able to answer those questions when you're sitting on such this?
Yeah, so there's 2 ways.
Forth for the tree.
Yes, so there's 2 ways to think about this. The first way, which I think is the most honest way, is most of today I've just been asking other professors, what are the biological questions that we should use this technique for? And I mean that in a serious way, right? Like I've been spending the last 7, 8, 9, 10 years thinking about how to make this thing work. And I haven't been thinking about what the biology would be. But now that people know the capabilities and the connectivity, that's one way. We ask people, and we then do the samples that they want us to do so we can answer the specific questions that they have. The second way is you put these data sets, as the data sets get larger and larger and larger, you should be able to then identify brain regions, brain locations, because now we've done 2 millimeters cubed or 3 millimeters cubed. And then you put it out there, and whoever works in that part of the brain can go through and analyze their connectivity and et cetera. The goal is never for me to analyze this stuff. I see my job as much more simple than that. I see my job as the guy who produces the data sets and then sends them out to the community. There's a few biological questions I'm interested in that we're going to try to answer with this, but I think the vast majority, I would hope, is driven by the community.
All right.
Thank you very much.